Hello everyone. In this video, we are going to see about blotting techniques. So what is blotting? Blots are techniques for transferring DNA and RNA and proteins onto a carrier so they can be separated and often follows the use of a gel electrophoresis. Types of blotting techniques. Blotting technique can be classified into three types. Southern blot, which is used to detect DNA. Northern blot is used to detect RNA. Western blot is used to detect protein. Southern blotting. This method involves separation, transfer and hybridization. The southern blot is used to detect the presence of a particular piece of DNA in a sample. The DNA detected can be a single gene or it can be part of a larger piece of DNA, such as a viral genome. The key to this method is hybridization. Hybridization is the process of forming a double-stranded DNA molecule between a single-stranded DNA probe and a single-stranded target patient or DNA. The principle: the mixture of molecules is separated. The molecules are immobilized on a matrix. The probe is added to the matrix to bind the molecules. Any unbound probes are then removed. The place where the probe is connected corresponds to the location of the immobilized target molecule. Steps in southern blotting technique. Genomic DNA. With the help of restriction endonuclease, it is divided into DNA fragments and gel electrophoresis plays the role long DNA fragments and short DNA fragments on agarose gel. Denatured by mild alkali for transfer and nitrous or nylon by the following DNA probe acts hybridized bands on the autoradiograph. Applications. Southern blots are used in gene discovery, mapping, evolution and development studies, diagnostics, and forensics. In regards to genetically modified organisms, southern blotting is used for testing to ensure that a particular section of DNA of unknown genetic sequence has been successfully incorporated in the genome of the host organism. Southern blots allow investigators to determine the molecular weight of a restriction fragment and to measure relative amounts in different samples. Southern blot is used to detect the presence of a particular bit of DNA in a sample, analyze the genetic patterns which appear in a person's DNA, analyze restriction, digestion, fragmentation of DNA or a biological sample. Northern blotting. Northern blotting is a technique for detection of a specific RNA sequences. Northern blotting was developed by James Alvin and George Stark at Stanford University and was named such by analogy to southern blotting. An outline of northern blotting: RNA extract, RNA bands, DNA probe hybridizes to RNA. These are the steps which are involved in northern blotting. Applications of northern blotting. A standard for the direct study of gene expression at the level of mRNA, detection of mRNA transcriptase, study RNA degradation, study RNA splicing, can detect alternatively spliced transcripts, study RNA half-life, study IREs, internal ribosomal entry site, to remove possibility of RNA digestion, often used to confirm and check transgenic. Western blotting. Western blotting is an immunoblotting technique which rely on the specificity of binding between a molecule of interest and a probe to allow detection of the molecule of interest in a mixture of many other similar molecules. In Western blotting, the molecule of interest is a protein and the probe is typically an antibody raised against the particular protein. The STS page technique is a prerequisite for Western blotting. Steps in Western blotting. A protein sample is subjected to electrophoresis on an SDS polyacrylamide gel. Electroblotting transfers the separated proteins from the gel to the surface of a nitrocellulose membrane. The blot is incubated with a generic protein, such as milk proteins or BSA, which binds to any remaining sticky places on the nitrocellulose. An antibody that is specific for the protein of interest is added to the nitrocellulose sheet and reacts with the antigen. Only the bond containing the protein of interest binds the antibody, forming a layer of antibody molecules. Applications. The confirmatory HIV test employs a Western blot to detect anti-HIV antibody in a human serum sample. Proteins from known HIV infected cells are separated and blotted on a membrane. Then the serum to be tested is applied in the primary antibody incubation step. Free antibody is washed away and a secondary anti-human antibody linked to an enzyme signal is added. The stained bands then indicate the proteins to which the patient's serum contains antibody. A Western blot is also used as the definitive test for bovine spongiform encephalopathy, referred to as cow disease. Some forms of Lyme disease testing unemploy Western blotting. Thank you.